To continue on the topic of enzymes, all right, last video, remember I said enzymes are proteins. Enzymes are also functioning as a catalyst in chemical reactions within the cell. So with that enzyme, being that it's a protein, being that it's a catalyst, there are three things that can change. The enzyme's ability to function, okay? And in terms of those three things, I briefly mentioned these when we talked about proteins, but things that can affect how an enzyme works are things like temperature, the pH, acid and base environment, and the salt concentration. And how those three factors, how temperature, how pH, how salt, change how an enzyme works is by denaturing the protein itself. So it takes that protein and unfolds that tertiary structure. Sometimes it'll also unfold the secondary structure, right? And what that does is that alters the three-dimensional shape of our enzyme. And now we don't have the nice little pocket that our substrate can sit in so that the enzyme can work, all right? So these are the three biggies as far as what the environmental things can impact how an enzyme works. How fast an enzyme works is going to be dictated by the amount of enzyme that's available, and also the amount of substrate. If there's a lot of substrate, very little enzyme, that enzyme is reusable, but it's got a lot of work to do to break down a large amount of substrate. If there's a lot of enzyme, but not a lot of substrate, that enzyme will break that substrate down very quickly. So how fast the reaction occurs depends more on the amount of enzyme and the amount of substrate out there. The ability of the enzyme to work at all has to do with temperature, pH, and salt. So when we're talking temperature, typically what we're talking about is heat more than cold, although cold can also impact how an enzyme functions. Heat is going to be what's more likely to denature the protein. Cold temperature is going to work a little bit differently. Cold is going to essentially, because it's cold, you're inhibiting how fast molecules are kind of moving around and interacting with each other. So in cold temperatures, that enzyme and substrate are moving more slowly, so they're less likely to find each other and interact for the chemical reaction to happen. Enzymes at cold temperatures will still function, but at a very inefficient level, all right? I say that kind of as a broad overview. There are some enzymes that won't function in cold temperatures, all right? But cold temperatures have the possibility for the enzyme to function, just to function much less effectively. Heat tends to denature the protein, and that's going to essentially cut off all of that chemical reaction because you don't have the protein there, okay? pH, whether it's acid or base, and salt also interfere with the three-dimensional shape of the protein, so they're going to denature it by essentially interfering with the hydrogen bonding that's holding the secondary and tertiary structure of that protein together, okay? So those are going to be some things that impact how enzymes work. The other factor that's going to impact how enzymes work are going to be competitors. So in terms of competitors, these tend to be proteins. They tend to be similar to the substrate, and they interfere with the enzyme and the substrate interacting with each other. All right, so I'm going to kind of draw this because I think it's a little easier to visualize. This is our enzyme. This is our substrate. Oh goodness, that's not spelled correctly at all. Substrate. There we go, right? Now, if we're talking about a competitive inhibitor, and I'm going to make it a different color. Is going to bind to the active site associated with the enzyme. All right? And I didn't talk about this in the last video, but I'm going to talk about it in this one. The active site is where the substrate binds to the enzyme. Okay? That's going to be the three-dimensional structure where the substrate and the enzyme interact with each other. That's the active site. So a competitive inhibitor binds directly to the active site. And the thing is, because it's shaped differently than the substrate, now what that does that enzyme can't let go of that competitive inhibitor. So essentially what it does functionally takes the enzyme out of production. It can't be reused. So the body can only have more of that particular chemical reaction happen if it makes more enzyme that can bind substrate instead of the competitive inhibitor. Okay? So the competitive inhibitor takes the place of the substrate. I'm going to draw this a little differently here. Maybe we should put that little bit in this one too. And we'll put it in this one. So I can talk about the non-competitive inhibition. Okay? Non-competitive, oh, what color do I use? Let's do orange. A non-competitive inhibitor is going to bind somewhere else on the protein. And when it binds somewhere else on the protein, what it does is it changes the shape of the active site. Now our substrate can't interact with the enzyme because it's not the right shape anymore. So essentially what it does, the non-competitive inhibitor destroys the active site. A competitive inhibitor binds directly to the active site, so the substrate can't bind to the enzyme. All right? And in terms of enzymes, where you may have seen some of this, all right? A lot of laundry detergents have enzymes added to them to break down proteins, to break down fats or lipids that are found in stains associated with your clothes. So if you get grass stains on your jeans, like when you were 5, or maybe more recently than that, that's okay too. There are enzymes that are added to help break down those stains so that they'll wash away in the laundry. All right? So that's kind of one application for enzymes. The other application of enzymes that you may be familiar with, a lot of pesticides that are used to kill insects and bugs, you know, because you don't want those crawling around your house. A lot of pesticides also are enzyme based, all right? And that's why pesticides can be harmful to your cat or your dog. Because if your cat or your dog goes over and messes with where you sprayed that pesticide, they can ingest some of that enzyme and it's going to shut down their cells instead of the insect cells. Okay? So pesticides typically are going to be enzymes, or more likely, enzyme inhibitors. All right, and if those enzyme inhibitors are there, whether it's a competitive inhibitor or a non-competitive inhibitor, it's going to prevent some chemical reaction from happening within that insect. And when you prevent basic chemical reactions from going in the body, whether it be an insect or a human, you're going to prevent certain products from being made, all right? And some of those products are important. Like if we have enzymes that inhibit cellular respiration. Cellular respiration is important for every single living creature. Insects, plants, all right, people, all undergo cellular respiration. So if you interfere with that process, you're interfering with the body's ability to make ATP. If you don't have ATP, you can't run the bulk of chemical reactions, okay? So essentially what happens is enzyme inhibitors can inhibit some metabolic process. That's how they function, okay? Specifically with pesticides, all right? I don't want to just say this is all negative, okay? Because in terms of that, where this is going to impact you as future healthcare providers, some drugs that are given or medicines that are given are enzyme inhibitors. Inhibitor. All right? If you've ever taken ibuprofen, ibuprofen is a competitive inhibitor, all right? And in terms of that ibuprofen, what it's doing is it's inhibiting the production of a certain protein within the body called prostaglandins. Those prostaglandins are associated with inflammation. When you've got inflammation, you've got some swelling. When you've got swelling, that impacts nerves. That's where you get the pain from, all right? So ibuprofen is an inhibitor. That's how you prevent or take care of headaches, which is one reason why you may take ibuprofen. So some medications are enzyme inhibitors. It's not just ibuprofen. You've got some blood pressure medications that are enzyme inhibitors. Some antidepressants are enzyme inhibitors. Antibiotics, a lot of times, are inhibiting various enzymes as well, okay? So in terms of those inhibitors, it's an important way to control chemical reactions within the body, all right? Whether it be a chemical reaction that you want to be lethal, like in the case of the pesticides, because you want to get rid of the bugs that are coming into your house, or if it's a medication you take, all right? Hopefully that made sense. If you have any questions, please let me know.